thanks a lot, Sharmistha, for agreeing second time for giving this talk, but on a different person, Obaid Siddiqi. Sharmistha is a faculty member at IIT Gandhinagar Biological Engineering and Biological Sciences. And she's going to talk today about Obaid Siddiqi, the father of modern Indian biology. Sharmistha, the stage is yours.
Thanks, Pradipto. I hope you can all hear me. I'm not very Zoom friendly. Okay, first of all, I hope everybody is safe wherever they are. We are going through extremely difficult times. And in fact, I would like to thank Sudipto and Michelle for giving me this opportunity. Initially, I was pretty daunted by the task of telling the story of Obaid to all of you. However, given how the newspapers are and how we are all distracted by all the bad news, this at least gave me a chance to escape from reality and kind of read about not just Obaid himself, but essentially the history of biology in India over the past 50, 60 years. So I would like to thank all of you, and I also thank the audience now for listening to what I myself have learned and been awed by in the past week or so when I have been preparing for this talk. Okay, so I'm assuming, I would like to thank the previous speaker, Professor Dotto, for a very enlightening talk about, and I really enjoyed. And I guess this is a nice transition because I think Professor Dotto is also from TIFR, and what we will be talking about today is extensively the TIFR story connected to Obaid and how biology came into TIFR and hence into India, modern biology. So with that introduction, I'd like to introduce all of you to Professor Obaid Siddiqi. He may not be as much of an iconic figure as Bose or Raman or P.C. Roy are, whose names we all know growing up. I myself am ashamed to say that I really didn't know his name till I started my BSc. And but he is in fact still considered to be the father of modern Indian biology. And I hope my story today to all of you will kind of explain why people call him that even now. So he has a very interesting, So Obaid has a very interesting life, not just his biological contributions. He has several different facets, and I will try to touch upon. It's very difficult to tell you the story in 1 hour, given that I myself am learning many things about him, and I personally never did know him. But it was fun finding out so many things about him. So just some historical facts. Obaid was born in 1932. This was pre-independence India, of course. But before I get into his life story, I'd just like to let you know what, in fact, makes him so famous. So, of course, he was one of the pioneering biologists of our time, not just in India, internationally. And what is fascinating about his story is his ability to kind of cross different literary kingdoms of life, if you will. So he started off as a plant biologist, biologist in India, and then he moved into fungal and bacterial genetics to learn what was then a very modern field of biology, molecular biology. And in doing so, he made some fundamental discoveries, and I will try to touch upon some of these scientific discoveries as we go along. One of them being the stop signals in the genetic code.
Sharmistha, you are mute now. Hello? Yeah, I think so.
Yeah, can you hear me now? Yeah, okay. And so moving from these fundamental molecular biology questions, he completely made an almost like a 180-degree shift into genetic neurobiology. And in the last 30 or so years of his life, he was working on chemosensation and in a model system, which is the fruit fly. So these were very fundamental pioneering work as a biologist, but that is just one facet of Obaid. He's, of course, most well known to the community, the scientific community in India, as an institution builder. Firstly, for starting the molecular biology unit in TIFR Mumbai, and then building this greater center, the National Center for Biological Sciences, which is now in Bangalore, and it's considered to be one of the, if not the best, one of the best places to do biology in India, and in fact is a world leader in certain subdisciplines. So just to tell you about his life, and what I decided to do is it's very hard to break this up, so I decided to just tell it chronologically, starting from his birth till 2013 when he passed away. So Obaid, like I said, was born in 1932, and he was born in Basti, which is in eastern Uttar Pradesh in pre-partition India. His father had kind of a job which made him move around UP there. So he grew up in Lucknow, Banaras, Allahabad, or different parts of UP, and finally the family settled in Aligarh. And that is where he continued his college education. So he did his BSc from, one second, I'm having some issues here. Is my screen still visible? Because I see some pop-ups here.
Yeah, the screen is visible. Yeah.
And you can hear me because there are some pop-ups coming. Okay.
I can hear you, and I can also see your cursor moving.
Okay. He was born in Basti, like I said, and then he continued to do his BSc in Aligarh Muslim University between 48 and 51. He went on to do his MSc in the same university in botany, and then he continued as a lecturer between 54 and 57 at the same university. All of this was in botany. So he was trained as a plant biologist in very traditional biology, traditional botany. And around this time, around 57, he decided he wanted to do more research rather than teach at this point of his life. And so he moved to New Delhi to the Indian Agricultural Research Institute in Pusa. This institute still exists. And he started working on wheat genetics. So this was agricultural research, and he was there for around a couple of years. And sometimes there are these interesting stories in science where you hear about something completely non-scientific, very circumstantial, which causes a person to completely change their path. So this happened to Obaid actually in this early point of his life. So it so happened that there was this weird hailstorm that happened in Delhi in 58. And for those of you who know agricultural research, crops like these kinds of food crops, it takes a long time to grow, several months. So if you want to do some kind of observational genetics, which was the kind of research people were doing in those days, you would need to really wait a year before you actually did your analysis on a certain crop. But this hailstorm, which was completely out of the blue, completely wiped out a year's research for Obaid in 1958. And this was, of course, very demoralizing for him because he was a young researcher. And his colleagues, of course, told him, yeah, this can happen. That's just the nature of the organism. You can just start again, and so on. However, in these intervening years, he was also starting to read about modern biology. So what is modern biology? For those of you who are not from the biology background, unfortunately, what we do in school even today is, in fact, this old traditional kind of biology. We don't really get introduced to much of modern biology in school. At least I never did get introduced. It was only in my bachelor's when I actually got exposed to modern biology. But when the modern biology term, kind of the umbrella term, essentially encompasses everything that is molecular about biology. And this kind of started in the early 1950s when DNA, the structure of DNA was discovered by Watson, Crick, and Franklin, and so on. And that kind of sparked this revolution, so to say, in understanding biology at that molecular level, and also appreciating that biology is not just this observational science where we just observe external characteristics, but there is a lot of things going on inside. And dare I say, there's a lot of physics and chemistry and maths involved in understanding those processes. And that is when this revolutionary thinking kind of started taking over the field. And of course, there was a lot of back and forth between the traditional biologists and the modern biologists. That's why I say modern biology. But in my mind, you all just call it biology, which is now how a lot of what departments are now renaming themselves. So there was a time over the past 50 years where a university would have a molecular biology and a botany department. But now more and more people are appreciating that there's no sense in kind of making these two departments. It's actually all just biology. And so we've come back to the boring naming of departments as life sciences or biological sciences and so on. So with that background, so of course, Obaid, this was early 1950s, and he was reading with a lot of fascination all that was happening in the biological world then, this DNA getting discovered, people understanding the basics of molecular biology, how DNA gets copied, how it is transcribed, and so on. And when this wheat crop of his got completely obliterated because of this hailstorm, he decided to make a switch. So a complete switch, actually. So what he started doing was, this was of course 50s, no email, no internet. People used to write old-fashioned letters. So he started writing letters to really well-known scientists across the world. This was again very common, even in those days, even without email. So you would just handwrite or type a letter and send it off by snail mail, what we call snail mail. And what he did was he sent letters around the world, but he finally got a response from the University of Glasgow. So, and that is when he decided to essentially cross the oceans. So he moved from not just India to the West, he also moved from being a botanist, a plant embryologist, regeneticist, to the molecular world of fungi and bacteria. So this is the year where he left the country, albeit it was actually quite a short visit, given how short PhDs and postdoctoral stints were in those days. So, so he kind of entered this world of molecular biology. And this is a quote which I thought I would share with you. So this is from one of the various articles I was reading about him, where I think Vijay Raghavan, and I'll come to Vijay Raghavan many more times during the talk, who was his PhD student, said that genetics, he decided to do genetics at a slightly faster pace than what wheat allowed. And that is essentially one of the beauties of these simpler life forms like fungi and bacteria, which can literally grow within a few hours, and you can actually follow generations of these organisms very, very quickly. And even if something was to get obliterated by a catastrophe like a hailstorm, you could quickly get back onto your feet within a day or two. And so that was a kind of a joke which people make even today about how that hailstorm literally changed Obaid's life. So anyway, coming back to his PhD, so he started doing his PhD at the University of Glasgow with, and I'm going to say this name wrong, Guido Pontecarvo, an Italian geneticist, fungal geneticist. And these are some pictures from those times. This is Obaid here sitting, and this is his supervisor. And what they were working on, this group over there, was fungal genetics. And there are a lot of interesting stories about how he finally got into this group. So it seems that he wrote a letter to Guido from India, and he responded saying, yeah, sure, you can join. And that was it. So there was, like all these formalities of today were not there. So what Obaid did was he just took off. He just took off and he reached Glasgow. And Guido was really surprised. And Obaid, on the other hand, thought that he was just being called for an interview, and asked him, so do I get to join you? And Guido said, no, your interview is your, the conviction that you have to leave India and come all the way. That itself convinces me that you are ready to do a PhD. So it's very different from the way we conduct interviews nowadays. However, it did really completely change both of these persons' lives. So Obaid started his PhD working on fungus, a completely different organism. The Latin name is here Aspergillus nudulans, in Guido's group. And what he started doing was essentially genetics, so understanding the structure of genes in this fungus. And Guido was a world expert in this fungus, specific fungus. This is the front page of his thesis, if you see on the left here. And I will not go into details of the work that he did there, but he did manage to publish a couple of papers in the 2 years that he took to do his PhD. And this paper, which was his most cited paper from his PhD, was in fact, it suggested introgenic recombination could be polarized. That is, when there is introgenic recombination, and there could be a marked concentration of mutational sites in one part of the gene. I won't go into the details of this research. I will explain some of his other more path-breaking work. This was definitely very fundamental, but there are some other pieces of Obaid's research which actually were completely opened new fields. So I would try to focus on those instead. Okay, so while he was in Glasgow, what happened was, of course, in spite of the fact that the world seemed to be was not as small as we think it is today, people were still traveling. So scientists would travel across continents, across oceans to meet each other, talk science. And there were several visitors in Glasgow, of which he actually got into several conversations with this person called Alan Garron, who at that time was working in Cold Spring Harbor, which is again a very iconic place in biological research in New York, and was just going to move into University of Pennsylvania. And this is a letter which Garen sent him. It's a little hard to read. These are from the archives. And you can see that he's essentially inviting him to come and join his lab for a few years and still remain in molecular genetics, study molecular biology, but in a different system, bacteria now. And so he's saying that I met you, I talked to you, and he was at that time still working at MIT and spending some time in Cold Spring Harbor, and he asked him to come over. So Obaid, of course, grabbed this opportunity. He decided to move to UPenn and with Garen. This is, of course, is a picture of a more recent picture of Garen. And what he did was he started working in bacterial genetics, and this is a place where he made one of his first fundamental discoveries. So Garen was, of course, a bacterial geneticist, and he went on to discover what are called the stop signals in a genetic message. And I'll explain what that means in a second. And what Obaid did in his lab was discover suppressors of these mutations in bacteria. This is a paper he published. But since this is a very general audience, I thought I'll explain what that means and why this is very, very fundamental. So just completely switching gears, I'll explain the science, the basics behind this discovery and why it's important. So even though this is a very general audience, I'm assuming most of you kind of know about the central dogma of molecular biology. It is, I say it within quotes because there are, as is common in biology, exceptions to the dogma. But by and large, this holds true. And this is one of the fact that DNA is, the DNA code gives, the message in the DNA is converted into an RNA message, which is then converted into a protein message. These basic steps of the central dogma are essentially conserved across the kingdoms of life. And this is one of the evidence, if you will, that we believe evolution really happened. The fact that across organisms, starting from the simplest bacteria to the most complex organisms that have lived or still live on Earth, all follow this DNA RNA protein paradigm. To explain this, I'll just kind of use an analogy from daily life. So again, this is something which you may have all heard. Was there a question? Hello? Is there a question or is somebody?
No, I think you proceed.
Okay. Okay.
And others are kindly requested to mute their mics. Sure. If there are questions, we can take them at the end.
Sure. Yeah. So just kind of giving a little background about what DNA is, given that this is a general audience. So I guess one thing that you always hear associated with DNA is the language of life or the blueprint of life, the genetic blueprint, if you will. So essentially, it is it's a code. It is a set of letters which tells your body what to do. Very, very simple English. And the analogy to a language, actually, I like very much because it's very easy to compare the language, say English, to the DNA language. And even the fact that we speak many languages. All of these analogies can be made with this DNA RNA protein central dogma. So just be with me. So the DNA, which is essentially a genetic material, this is present in every single cell of your body. So we are multicellular. We as humans, we have trillions of cells in our body. And all the cells in our body have the same DNA, more or less. So that is the blueprint that is present in every cell. And that is and it has 4 letters. This DNA language that I'm talking about, unlike the English language, which has 26 letters, the DNA language is 4 letters. Very simple, if you will. A, E, G, and C. Which you may have heard, the ATGC language of DNA. And this ATGC language is then converted, so it's translated, which is why it's called translation. So this DNA language carries the code. It is the code which is carrying the information for synthesizing a protein, hence it's a language. And this process of converting this DNA language into a protein language is called translation. It is translation, given this analogy of the language. So just like I'm speaking in English now, I also speak Bangla, which is my mother tongue, and I try to speak Hindi, which I've kind of learned off the cuff. And if I was to speak if I were to speak in English, I also speak in Bangla. And if somebody else I'm speaking to doesn't understand either of these languages, we would require an interpreter, right, to understand my language, the English language, and then translate it into, say, Bangla, and then convert it into Bangla. So if there are 2 people, one speaks English, one speaks Bangla, they don't speak each other's language. You need an intermediate person who speaks both so that they can translate. That's the interpreter's role. And that is what RNA does. So the mRNA actually reads the DNA language, it is the same language, and then translates it into the protein language. So that's essentially what is going on. That is the central dogma of biology, kind of narrowed down to these individual letters. Of course, there is much more going on within a living system, but at that molecular level, that is essentially what is going on. It's conversion of language. And so all of this is well and good, right? So, okay, so DNA language gets converted into RNA language, the RNA language gets translated into protein language, and that is how the cell understands what to do in very simple terms. But a more fundamental questions, when you're writing English in the English language, we write sentences and words, right? So very similarly in the DNA language, you can have words. And these words are of course much simpler given that they have just 4 letters. English has 26 letters, DNA just has 4 letters. So there are only so many combinations of words that you can get. And another constraint is that the words in the DNA language are always 3 letters long. So these are called codons. So this is what the genetic code is, as you can see over here, and I'll come to that in a second. But even before that, one of the other important questions is how do you even know what is the beginning and the end of this sequence that is encoding for a message? So in an analogy to the English language, a capital letter defines the beginning of a sentence, a full stop defines the end, right? So, but is there any similar thing going on in the DNA language, given that at the molecular level, there is no human being reading, right? It's other molecules reading and translating. So how is it able to recognize that this is the beginning and this is the end of the message? So it turns out very, very fascinatingly that there is a certain code, a 3-letter code, which is coding for what is called the start of a message, and another set of three 3-letter codes, which essentially signify the stop of a code. And this is in fact the genetic code, which I won't go into in a lot of detail. But if you look at these 3-letter possible combinations of the DNA language, there are 64 possible combinations of words which come from the DNA alphabet. And of these 64 possible combinations, this 3-letter code marked in red is the start codon, which is like the capital letter in an English sentence. And these 3-letter codes, which is UAA, UAG, and UGA, these 3 codons is what we call it in biology, are called the stop codons. Okay, so they define the end of a message. And that is how the cellular machinery understands that, okay, I need to start translating here and stop translating here. And that is the beginning and end of the message. I would also like to remind you that I'm just talking about one single gene. And we have approximately 30,000 modes of genes, at least in the human genome. And depending on the organism, you can have more or less genes. So that's for another day. So why did I tell you this whole story? Molecular biology dogma, start codon, stop codon, and so on. So where does Obaid fit into all this? So when Obaid started his PhD, none of this was known. DNA structure was discovered, but all of these other steps that I discovered, which we take for granted, are in all textbooks today, was not known. In fact, that is why we call this the age of biology, because all of these discoveries are being made as we speak. So we literally watched these discoveries over the past 50, 60 years explaining biology at that molecular, submolecular level. So Obaid, when he did his postdoc in UPenn, just this fundamental important piece of information, how does a DNA message start, how does it stop, was not known. And what he did in his postdoc, which is what I explained here, was he discovered suppressors in bacteria. He discovered certain signals in bacteria which were suppressing the stopping of a signal. So he was making mutations in bacterial genes, and he found that he made certain mutations, and the reading of the message did not stop. So essentially, inadvertently, he discovered that there were certain stop signals in the code, which when suppressed, prevented this translation stopping. And so he, of course, left the lab around 62, but Al Garin went on to characterize these further, and this led to the discovery of these stop codons in bacteria at that point, but then we now know that this is a universal truth. It's one of those things which is conserved across evolution, starting from bacteria, even in human genome. The stop code, the stop signal for a message is always these three three-letter words, which are called the stop codons. So in that sense, this was Obaid's first connection with a very, very fundamental biological discovery, which kind of set the stage for Al Garin to do more elegant experiments in the molecules and able to discover stop codons, which now turn out to be one of the fundamental truths, if you will, of biological molecular biology. Okay, so this was his postdoc work, 1962. So if all of that science was a little heavy for you, we'll switch gears and we'll come back to reality. So in 62, after he published these papers, and of course, you would notice how these stints, his PhD and postdoc stints, were much, much shorter than how they are today, 2 years, 1 year. But it was also the nature of in those days. So around 62, when he published these papers, Obaid decided that it was time for him to come back. He always actually wanted to move back to India. And this was not because he was not getting a job in the US. He had several offers. And in fact, as I go along this talk, you realize that he knew literally the who's who of biology. So when he was in the US, he was hanging out in Cold Spring Harbor, where a lot of these biology workshops are still held, in fact, to this day. And that is essentially a place, it's kind of one of these pilgrimages of biology, where all the stalwarts of the day kind of congregate over the summers to have these very specialized, advanced, short training programs on modern topics in biology. And he used to spend a lot of time in Cold Spring Harbor, and he met and knew many people, including Watson Crick, the discoverers of DNA, and Brenner, Benzer, Morris Cox, all of these people who he kept connections with till the end of his life. But in spite of his connections in the US and him doing so well, working literally at the cutting edge of biology, he always did want to come back to India. And so what he did was around 62, he started exploring. And so just like he was writing letters to go to the West to learn molecular biology, he again did what everybody did in those days. He started typing letters. And he sent letters for feelers to several of these, the top scientists in India, who in fact at that point, many of these iconic scientists were of course from physics. They were all physicists. And he did write to, in fact, almost all of them. So this is a copy of the letter he wrote to S.N. Bose, who was in Calcutta at that time. And Bose, in fact, responded. This is Bose's response to Siddiqui's letter, telling him that, yeah, great, this is, I know this is an upcoming field of biology, and you should definitely come and set it up in India. And he's giving him suggestions of places where he can apply to. He suggests many of these national institutes in Delhi. And he also tells him that, be in touch. If I hear of something, I'll let you know. This you can see is 20th December 1961. So he had started exploring. He also had talked to his US contacts, telling them that he wanted to return and he was looking for places. So meanwhile, what happened was, and this is where the story gets very interesting, where the Bhabha and TIFR connection started developing. And this was in fact a very interesting connection. So with S.N. Bose, the letter I just showed you, he himself had written to S.N. Bose. S.N. Bose did not write to him. He didn't know Obaid. He didn't know who Obaid was. He was this young researcher working in Pennsylvania. On the other hand, and I'll give you a little background about Bhabha, since we will be essentially kind of living in TIFR for the next several slides. So I guess to this audience, I don't really need to introduce Bhabha to all of you. However, just for the sake of completeness, I will. So Bhabha, of course, is considered the father of the nuclear program in India. And in addition to being a great scientist, a nuclear physicist who was trained in Cambridge, he is, of course, best known for starting the TIFR, the Tata Institute for Fundamental Research in Mumbai. And some of you may even know the story about how he was working in Cambridge. During the Second World War, he came back to India for a short trip, hoping to return. However, he got stuck. I think he started working in IST, and I may be getting some facts wrong. And then I think since he could not return, he decided to stay back in India. But he also decided to set up a center of fundamental research, which is how the TIFR came about. And he directly wrote to the Prime Minister Nehru and so on, was able to get funding from the Tata Trust, and of course the rest is history. So this is early 1960s. So TIFR was functioning in Mumbai in the Colaba campus, and of course it was well known for theoretical physics. That is what, and mathematics a little bit at that point. And Bhabha was there, and so why was Obaid even trying to contact these physicists, you may ask? If you think about it, physics and biology, even today we consider ourselves to be the opposite ends of the sciences, although more and more you see people intermingling. It's not, and that's just a joke. However, Bhabha, of course, was sitting in TIFR. And what happened at this point when Obaid in Pennsylvania, he started talking to, like I said, he was talking to his supervisors. And his supervisors, again, knew several other scientists in India, in the US. One of them being Leo Szilard, who was one of these very well-known physicists, not just for his physics. He was a physicist who later turned to biology in the later part of his life. And he's very well known for his involvement in the Manhattan Project, and later being involved in nuclear disarmament and so on. And he was a Hungarian-American physicist, and he's made several discoveries in nuclear physics as well. So Leo Szilard knew Garin, who was Obaid's advisor, and he heard that he was this really promising guy who should actually move back to India and would do great things in India. And so Szilard, what he did was, okay, I've heard of Bhabha. I don't know him directly. I'll write to Bhabha, and Bhabha will probably notice my letter over a letter written by this unknown young biologist. So Szilard, who really didn't know Obaid directly in terms of working with him, but via Garin and via seeing him working in Garin's lab, wrote this letter, and I'm actually directly writing from his letter, that he actually sent a letter. This is a very unique application. So there was no application form, of course, in those days. This was a letter from Szilard with 2 recommendation letters. One from the postdoc advisor, which was Guido, the PhD's advisor in Glasgow, and one was from Garin in UPenn, and it was forwarded by Szilard, who was his famous physicist. And it said that I enclose this letter, which has 2 recommendation letters, and I should be grateful to you for reading these and following it up. It's appropriate, and I regret that our paths haven't crossed. So this is Bhabha, Szilard to Bhabha. And then Pontecarvo also said it is important for the progress of biology in India that he should go back. And I'm really baffled. This is a very interesting line which I noticed. I'm really baffled as to why India continues to promote mediocre scientists, politicians, and does nothing to maintain the really good scientists. This Pontecarvo tells Bhabha in 1962. And in fact, Bhabha really was caught by this line as well. And in fact, he actually took it to Delhi and made PM Nehru read it as well. He said, see, this is what people think about Indian science, and we really need to fix it. And we really need to create these institutions where fundamental science is done, and we don't have scientist politicians. We have really good scientists doing fundamental work. So after reading all these letters, Bhabha was, of course, really intrigued. He just wanted to find out who this guy was, this young guy who all these letters were coming for, who he had never met or heard of. So he himself wrote to Siddiqui. So he wrote to Siddiqui saying that I received this letter. These are all actually, I'm actually quoting directly from their letters. These letters are all available in the NCPS archives. And I'm very interested, and this is actually, you should know that this is coming from the TIFR, which at that time was a world famous theoretical physics mathematics institute. There was no biology, and I guess most people didn't even know what molecular or modern biology was. But Bhabha, of course, given who Bhabha was, had heard about molecular biology and its advances, and he in fact directly wrote to Siddiqui saying, I would like to support somebody in molecular biology, and I think we can give you an offer at TIFR or the Atomic Energy Establishment, which is I think the Park Center, and why don't you send your CV now? I've already got your record letters. So it's kind of a very unique kind of application, very almost unheard of nowadays. So of course, Siddiqui naturally complied. He did send it back. He did discuss this with many of his other friends in the US, and several people in fact told him that in fact, this is a great that you're being approached by a physics institute and not a traditional bio institute, because given the times where molecular biology was emerging as this new field, a lot of his friends in the US thought that he would get more resistance from the traditional bio institutes which were doing traditional botany, zoology research, compared to a physics institute where he would just literally be left alone. And so they actually said, oh, this is great. Why don't you just take this up? If TIFR is giving you a job, definitely look into it. So Siddiqui sent back his CV and so on, and Bhabha gave him an appointment. And this is another fun fact. So Bhabha gave this appointment to Siddiqui without actually consulting any of the faculty at TIFR. What he did was there was a faculty meeting in '62 with the faculty of TIFR, and Bhabha then went and asked them, what do you think of hiring somebody in modern molecular biology? And the funny part is almost all the faculty sitting in the room said, no, we don't want a biologist in TIFR. We are a theoretical physics institute. We don't want biology here. Bhabha just listened to them and said, hard luck, he's coming next week. So it's one of those things where, an autocratic, so if you will, decision by a director creates history. And this is one of those examples. And he went on, in fact, in that same meeting to ask the faculty in TIFR what kind of furniture they could share when Obaid arrives later in 1962. And this is the memo which I talk about, which I stick. And if you're interested, you can read this later. It gives more history of Obaid, what he's doing, and what he's going to do here. So anyway, so to cut a long story short, that's how Obaid reached Mumbai. So so at that point, it was not a department. The department came much later, which still existed, the Department of Biological Sciences, DBS, in TIFR Mumbai. But in 1962, to accommodate Obaid, they called this a unit, MBU, Molecular Biology Unit. And what he got was, in terms of space, was this one big shared lab. Which was, and wherein he was allowed to recruit several faculty. He was given full freedom, of course, a lot of funding as well. And this, in fact, grew into one of these essentially hotbeds of molecular biology research in India. So I would like to point out, and this is something which many people still reminisce, people who worked and went through that lab system. This was one of those first open shared labs in India. And I guess, not to brag, but what we have in our campus also is kind of following the same philosophy, wherein we have one large space, open, shared, where there can be free exchange of ideas and materials. That's the idea. That's the general philosophy behind it. Of course, it can lead to a lot of conflicts as well, but then people manage to also learn how to deal with such conflicts, given if you have so many people in a place. It's like an Indian joint family system. And the mandate that they had was very simple, the mandate of molecular biology, essentially. So the mandate was to explore all forms of living organisms in terms of their, at the molecular level, as you can see. So in terms of molecular reactions, without these old-fashioned boundaries that existed in botany and zoology. So you understand everything at the molecular level, be it bacteria, be it yeast, be it worms, and so on. And that has continued to be the mandate of the TIFR BBS, which is this current thing. So at this point, I thought there's actually a lot of resources on Obaid and his history on the internet. And I found this very interesting YouTube video, which TIFR came up with on Bhabha's life. And within that video, there is this short snippet where Obaid himself tells us about how he started this MBU at TIFR. So I'd like to play it. It's a short 1 minute.
Those were the days in the 50s when I wanted to come back to India, and molecular biology was growing. 1953 DNA was discovered, and those 10 years between 53 and 62 were the ones in which most of the ideas of molecular biology came into fruition. My first meeting with Dr. Bhabha was in his office in the old yacht club. He asked me whether I knew what my requirements were. And I said to him, if you tell me what are the sorts of limits within which I have to make my plans. And he laughed and he said, when you begin to cross the limits, then I will have to call you and tell you that. So that sort of put me at ease. Most of the institute building had been built by physicists. So there were no provisions for sinks and fume hoods, the kind of facilities that biologists need to work in the lab, what we call the wet lab facilities. When Dr. Bhabha saw that the only way we could have fume hoods in the main buildings of the institute would be if we cut the windows and had the ducts passing through the windows, and he didn't like that idea at all. So he said to me, well, we might better build a new laboratory for you. So he asked me to make plans for the lab and make drawings, which is what I did for the next 6 months. And he didn't like the drawings at all because he said that I had made them very crowded. And he took it home, and a couple of weeks later he called me and gave the drawing back to me and said to me, "Don't you think it looks much better now?"
Impossible task was something which I think Obaid has taught us to do. If one looks logically at what is possible today, there's very little one can do. All the odds are stacked against anything you want to do in science here, in institutional building here, in terms of impact on society here. Yet, embarking on that direction changes the goals dramatically. Not embarking is certain defeat, but embarking makes it actually possible and successful. And that's what I think both in Bombay at the Molecular Biology Unit as an institutional builder there and at NCBS, Obaid said that, let's embark on new kinds of odds. And it is to his credit that he was not defining what those directions should be, but always supported you after the event, no matter how heated discussions were before. If you needed help, he was there. And if things worked out, he acknowledged that, those directions which he earlier felt were not the wisest thing to do were fine. And if you actually failed, he was always there to help you. Whenever there was a heated argument, he was the first one to come to you as an elder and to actually reset the discussion rather than you doing it. And that attitude, that culture is something which I think we have in this context. Some institutions in India have it, but the more one has that ability to actually have heated discussions, disagree, and come back, the more successful we'll be as institutions. Thank you.
Were you able to watch the video or hear the video? I think watching was not a problem.
Only a few seconds. Yeah.
And then it stopped again. Okay, so I'll just tell you, essentially just Obaid himself speaks about how Bhabha invited him and then told him to look for space, design lab furniture, and he came up with some lab furniture, some lab designs, and then Bhabha said, this looks very crowded, and it's a kind of this fun back and forth between him and Bhabha, and how he finally then, so he actually shows, in fact, these are from his notebooks from the time. So Obaid actually, this is handwritten notes of furniture design for the new MBU labs in TIFR. You can see how he's drawn lab benches and cabinets and office desks and so on. Gave these to Bhabha. Bhabha spent 2 weeks, made them much better. And then that is how the first lab came about. So that was kind of Obaid reminiscing about Bhabha setting up the MBU unit. Well, I'm sorry for those technical difficulties, and I'll share the link with all of you. Okay. So this was how MBU started, the open shared labs in MBU, and over the years, starting from the early 1960s, Obaid was instrumental in recruiting several very interesting biologists from all kinds of genetics disciplines, from yeast genetics to worm genetics, bacterial genetics, and of course, he was also doing his own science. So it's not like he was just institution building. While all of this was going on, he was setting up lab in this physics institution, building, making furniture, making labs, and so on. One interesting thing, which in fact the video I was just showing talks about, was how even these basic requirements for a wet lab were not there in TIFR, like for fume hoods and for water. The spaces that he was given did not have the provision for setting these up. So he actually had to go out of his way to make these, and Bhabha and he went back and forth about how certain designs were uglier and so they would have to be redone. But eventually they had a working lab. And in spite of all of these other distractions, he was still able to continue his research. And he, in fact, continued his research till essentially his last day. He lived till he was 81, and he was working on the bench even on that day. So he had still continued his work in bacterial genetics. And at this point, he was working on what is called conjugation. Conjugation is the ability of bacteria to transfer DNA without mating. So like wherein there is it's not the kind of exchange that happens during sexual reproduction in higher organisms such as us. So it is without DNA replication. He was able to establish this in E. coli, and he was one of the many people in the world who were working there on conjugation. And he published several papers, some of which I have listed here. And he was still very much in touch with the bigger world, in fact. So throughout the 70s, 60s, he would keep going back to the US. He still maintained his connections. And in 1970, he in fact was a Ford Foundation professor in Maury Cox's lab in MIT. This is a friendship which lasted their entire lives. And he went back, and this was a time when not just the biology, but the techniques to do this biology were also evolving very, very rapidly. So he went to Maury's lab, he picked up a lot of techniques, came back, and that's when he started working on conjugation, the ability of bacterial cells to exchange DNA via what is called conjugation. And these are some of the papers that he published in those years, in the early 70s. Okay. And of course, everything was going well, and you would think that, okay, he was now a little over 40. He had established this unit, recruited great people, set up his lab. His family was also growing at the same time. So I didn't really get a chance to talk about his family, and probably won't, but he was actually married to a very illustrious lady, Asiya, who was a very well-known historian who just passed away probably a year ago. And they had 4 children, all of whom are also very well established now, mostly in academics. And so he had a young family, he was really busy, but then he was getting bored and he wanted to move into this other new area. So in 1970, so this was when he was around 40, he decided to move from his comfort zone of fungal and bacterial genetics to something completely different, to neurogenetics. So what is neurogenetics? This was a completely new field at that point. It was even almost iconoclastic, if you will. It was kind of not accepted by the traditionalists in the early 70s. So this is, of course, very well accepted now. But at that point, this scientists, a few scientists started suggesting that a single gene mutation approach, it was the approach that people were using in fungal and bacterial genetics, that a single gene can be attributed to some kind of function. So this gene function approach, you can use this. And so to study this, very common method, which we even still use today, is we hypothesize that a single gene can be attributed to some kind of physiological function. And the best way to understand if our hypothesis is correct or not is we mutate, which is essentially we kind of try to make that gene dysfunctional. So we try to change the sequence of the gene. So I just talked about the DNA sequence a little bit earlier. So there are ways to manipulate that DNA sequence. You change it and see if that function that we think it is responsible for is still there or not in the organism. And that is how you can go back and forth and come up with connections of a gene being responsible for a certain function. In the 70s, this was not accepted. People said, yeah, you can do this in bacteria and simple single-celled organisms like bacteria, some fungi, and so on. But to understand the nervous system in more complex eukaryotes, the nervous system that we all have that allows us to perceive, to behave, to sense, genetics cannot be used to study something that complex. But there was a small group of scientists who started saying that no, that depending on the system, we can use genetics to understand something at that very, very, very basic, that molecular level, understanding a single gene approach to understand the nervous system. And so of the few people in the world who were thinking about this, one of them happened to be a very good friend of Obaid. This was Seymour Benzer. He was also, and I guess many of you probably know this, that at that point of time when biology was kind of becoming modern, many of these new age biologists of that time were actually originally physicists who then turned to biology. Francis and Crick and Watson were both physicists. Rosalind Franklin was a physicist. Benzer also was a physicist. He was a physicist at Caltech who actually decided to study understand genetics, neurogenetics, the nervous system, neurobiology, and make this connection with genetics. And something which again a non-biologist may not know is that you will see that there are certain, like in biology and even today, we have these model systems. What are model systems? So model systems are certain animals which scientists take up as a model to understand biological phenomenon. And given that you're using it as a model, there are a lot of techniques developed around that model to be able to understand it better. And the fruit fly, the Drosophila, which you see over here, is one such model system, which is like one of the most popular model systems out there. And this started coming about in the 60s, 70s era. And Benzer was one of the first people who decided to study the nervous system using the fruit fly as a model. Obaid heard about this. He, of course, was corresponding with all his buddies in the US. And Benzer said, why don't you come over and let's just, talk about these ideas, work on some ideas together. So he went on a sabbatical to Caltech. This was 1971. And this is when, so these are some pictures from that time. This is Benzer and Obaid chatting. This is him, Obaid here, sitting with the rest of, this is Obaid over here. This is the rest of the Caltech lab. And this is, of course, the famous model system. And starting 1970, like Obaid was hooked to the fruit fly. This is something which is a joke in biology, that once you start working with the fruit fly, you never change, never shift. This is a Drosophila geneticist joke, and Obaid is one of those people. So starting 1970, he stopped working on any other organism. He did look at several different questions, but he still kept using the Drosophila as a model until his last day, actually, his last day on Earth. But he got introduced to this organism well into his 40s, as this shows. So what did they start looking at? Of course, Drosophila they made friends with, but what were they looking at in the fruit fly? So in Caltech, the question that they decided to ask, this was absolute beginning days of genetics, using this neurobiology with this genetic approach. So they decided to look for genes which affect generation and transmission of electrical signals in the nervous system. Very simple question. People appreciated, of course, even then, that there are electrical signals which are essentially transmitting all information through the nervous system. The signals are getting transmitted. These are electrical signals, and that's true for all organisms, including us. But are there any genes which affect this? Very simple question. Again, now it is completely accepted. Neurogenetics is a very accepted field. However, at that time, this was kind of this question was almost laughed at by traditionalists in the field, traditional neuroscientists of the field. So what they did was, again, they did what all geneticists do. They started making mutants. So they started changing parts of the DNA of these flies to see if it could be connected with some kind of phenotype. Phenotype is some kind of outward manifestation, which would imply that the nervous system is affected. So they did series of experiments, and what they were able to identify, 3 genes, and Drosophila scientists are known for naming genes with these crazy names, and so even Obaid and Benzer did that. So they call them comatose, para, and sh, which are these three flies, which had this very interesting thing going on. So they made some mutations in certain genes, and when these genes were mutated in the fruit fly, what happened was they were abruptly paralyzed when they were shifted to high temperatures. So at normal temperatures, they were completely fine, normal as in room temperature. However, if you shifted these flies to slightly higher temperature, they were completely paralyzed. So what they were able to identify at the molecular level were 3 genes which were temperature sensitive, and what happened was at high temperatures, these flies were not able to move. So there was some kind of connection between temperature sensitivity, movement, and these 3 specific genes. So I would like you guys to kind of think just about one more time about this. So there are an organism usually has thousands of genes. We have many thousands of genes, and paralysis or temperature sensitivity is a very broad biological function, right, if you will. But of these thousands of genes, they were able to identify just 3 genes which were responsible for this happening. And this was almost revolutionary at that time, being able to connect a nervous system response to 3 genes. And so they call these the temperature sensitive paralytic mutants. And of course, it did several elegant studies to understand what was going on and the exact mechanism. And they were also able to show that this was not an irreversible process. So if they moved these paralyzed flies back to room temperature, they were again able to move. So something was happening which was temperature sensitive and which was connected to movement, which again was connected to 3 genes at the DNA level in the genetic blueprint. So in addition to, of course, this very fundamental discovery that you can have this neurogenetic discovery of connecting genes to a nervous system response, while doing this, this was of course published in PNAS in 76. This is Obaid and Benzer's paper, Neurophysiological Defects in Temperature Sensitive Paralytic. This came out of his sabbatical. But while studying this, given this was a completely brand new field, they also started developing these methods, literally experimental methods, because this was something which no one was doing before. Like flies are really, really tiny. So fruit flies are the flies which come and sit on rotting fruit. That's why they're called fruit flies. So now that it's hot, if you leave out a banana or any fruit outside, you always have these little flies sitting on them, right? these boring little flies that you want to get rid of. Those are fruit flies. And those have given us millions of discoveries over the past 60, 70 years. So they are kind of one of those models in the biological sciences. So develop methods to study these tiny little flies is difficult, you can imagine. These are really tiny. Of course, they're bigger than bacteria, so you can see them. So what, in fact, Obaid, Benzer, and several other of these neurogeneticists, new breed of neurogeneticists started doing was creating methods to do recordings on these flies. And so what they came up with were these very elegant techniques to record electrical recordings of single muscle fibers in these flies to identify and understand the mechanism. They also did a lot of these, and also this method of shifting temperature and figuring out changing of a behavior was something also that was very unique, which they were able to identify. And these, in fact, are still used even to this day. How they did these recordings, using temperature sensitivity as a way to study nervous behavior is still used even to this date. So in addition to the fundamental discovery, the techniques also were very important contributions at this point of time. So with this, of course, Obaid moved back to India. So he had just gone for a sabbatical. And during these times, of course, he made several notes. This is a notebook from, again, the archives. And it has a very interesting title. It's called Ideas for Experiments, Media and Solutions. And it is there are several pages of this which are scanned and on the NCBS archives. But I thought I'll just show you some of these pages where he designs different machines, which he actually built with his own hands, to be able to make measurements on these fruit flies. And some of these are still in the NCBS archived museum archives if you visit the Bangalore campus. This is one such thing which he built. And in fact, several of these were used to do these experiments. So when he came back to Bombay after his stint at Caltech, the sabbatical stint at Caltech, and now that he was familiar with working with this fruit fly model, he started looking at different questions. He moved away from movement and transmission of electrical signals for movement to this other field, which again is a very fascinating field, still very relevant today, which is chemosensory neurogenetics. So it's kind of a subfield of neurogenetics. So how do we sense taste and smell? How does our brain sense taste and smell? So this is something which we take for granted. Of course, we also have very complex regulation of our to understand the sense of taste and smell. And what we do have is that, for instance, for taste, on our tongue, there are several what are called sensory receptors, which have very high specificity for certain tastes. So there are some which can recognize sugar taste, some which can recognize salt taste, and the information is sent back to the brain, the electrical signal, and it tells you that you're eating something which is sweet or sour or salty and so on. And so in the 70s, using these techniques that he developed in the Drosophila fruit fly, for the next several years, he almost completely worked on chemosensory neurogenetics, wherein he used these neurogenetic techniques to understand very specifically how the Drosophila is sensing smell and tasting different kinds of flavors. And this is the time when many of these iconic scientists joined his lab. So several of you may have heard about Veronica Rodriguez, who actually was one of Obaid's Ph.D. students in the 70s. And she continued after she finished her Ph.D., she continued as a faculty there, but she unfortunately passed away almost 10 years ago, pretty early of cancer. And another person who I think all of you know is K. Vijay Raghavan, who is currently the principal scientific advisor, PSA, to Government of India. He was also Obaid's PhD student in the late 70s. He was in fact IIT Kanpur chemical engineer who was fascinated by Obaid's biology. And in fact, I have a letter from Vijay later on in the talk, which you can read later on, telling the story about how he decided to join Obaid for his PhD and so on. And during this time, they all worked on several different genes which were involved in taste and smell recognition. And these are some of the papers from that time. But what I wanted to highlight, not to say that papers in Nature and Science and Cell are the only good papers. There were several years of very interesting work. But one of these papers I thought I would highlight, which was one of Veronica's papers from '87, Veronica and Obaid, published from the TIFR, where the reason I chose this was not because it's in Nature, but because in fact it would be easy to explain it to all of you, given that I think all of us are familiar with the concept of taste. So unlike us, we have tongues to taste our food. Drosophila are very fascinating. They have sensory neurons, which sensory neurons are essentially nerves which can sense taste all over their body. So they in fact literally taste a fruit if it's sitting on rotting fruit by neurons on its wings, on its legs, and also on this proboscis, which is kind of this long snout-like thing which it has at the end of its head, kind of where our nose is. And all of these have neurons which allow it to taste food. And although it's such a tiny little organism, it has a very, very intricate taste system. It has different neurons which can sense salt, sweet, even water. It has a neuron which specifically senses the presence of water in some kind of food, if you give it. And these have been actually, people are still, this is a very active field. People still work on it. There have been Nobel prizes given to scientists who have elucidated the ability of an organism to sense taste. And most of these people were initially started their work in Drosophila and moved on to other organisms, including human. And it's amazing how conserved some of these taste pathways are across the kingdom of life. Okay, so coming back to the paper. So this paper from Veronica, Kavita, and Obaid, which was published in Nature in 1987, actually was kind of unique at that time because what it did was so these taste sensory neurons in Drosophila, they actually identified one specific gene, mutating which the taste sensitivity switched. So this neuron, which was usually sensitive to only sugar taste and aversive to salt, switched its essentially loyalty. So instead of liking sugar and hating salt, it started liking salt and hating sugar. So somehow this mutation changed the wiring of the brain literally, because these signals are all going back to the Drosophila brain, and prevented the Drosophila from being sensitive to the, essentially from being averse to the salt taste. And this was the first instance of a mutation which changes the specificity of a chemosensory neuron, which is why probably it went into Nature. This was this first example, and hence pretty pathbreaking. And of course, like I said, over the years, several other such genes have been identified. And in that sense, what happened was, because of Obaid's shift and now continuous success in this chemosensory neurogenetics field, he was, at that point of time, India was well known as one of the centers for this field of research in the fruit fly model. In the late 90s, he also started using the fruit fly to go a little more deeper into neurobiology and explore the nuances of behavior. And he did a lot of very, very, very cool, elegant studies where he actually studied the neuronal responses of single neurons using these machines that they built. And of course, technology also advanced over the next 20, 30 years. And at the level of the receptor, at the level of the brain. And these are several papers which came out over the years. And you can see how he kept publishing till the very end. He passed away in 2013, and there were papers which were coming out even then. He was over 18 at that point of time. Okay. So that's all I have to tell you about his science. And what I have not told you yet, and I guess we did kind of get delayed because of all the video drama, is I will kind of shift and I'll tell you a little bit more about Obaid as a person. Oh yeah, and before I do that, these are some pictures from those days. So this is Obaid with his own self-assembled electrophysiology rig in the 70s. So he actually made this machine out of different kinds of parts which he purchased in the Mumbai market to be able to do these recordings on flies. And these are his groups from those time. If you've seen Vijay Raghavan, you can probably recognize him. He's sitting right here. This is Vijay Raghavan. This is Obaid. And this is Veronica sitting next to him. And these are other famous people, many of whom have gone on to become leaders in their own right. This is a picture much later from the 2000s. If you see here, this is Taslim. He's currently the director of CCAMP. Sitting here, he was also working with him in the early 2000s and so on. And then, yeah. Okay, so around this time, of course, he was not just doing science. He never just did science. He was essentially what is called a Renaissance man. He was interested in several different things, and I'll come to that right at the end. But while all this exciting science was going on in the TIFR Mumbai campus, what Obaid and others were also appreciating was that they lacked space. And if you've gone to TIFR, you realize that. Like even now, of course, TIFR has expanded to other departments. There are separate departments. The MBU is now called the Department of Biological Sciences, but they do have less space. And Obaid felt that for biology, the way it's growing, we need a separate national center. And so he went with these proposals starting the late 70s and early 80s with a proposal to build a separate center for fundamental research in biological sciences. And to do this, he had several buddies, including Professor Vidyanand Nanjundia, who some of us have met when he came to our campus around 2 years ago. And these people were instrumental in coming up with these proposals. But what is frustrating as well as admirable about this story of how the Mumbai, like how NCBS came about, which was kind of the second phase of his institution building career, was the patience that was involved. So the idea came up in the late 70s, early 80s, when all of these people proposed this. It went through several rounds of iteration with the Atomic Energy Commission, Planning Commission. They spent several years looking for land in Maharashtra and MP, finally settled down on Karnataka, and actually over 10 years. And finally, when nothing was moving, there was again this IIC connection when and this is an interesting story for those of you who are from physics. So I guess all of you know about the giant meter wave radio telescope in Pune. So it turns out that the IIC campus has a small unit, which is called the TIFR Center on the IIC campus. I don't know if it still exists, it probably does. And the Govind Swaroop's group was actually there when they were setting up this GMRT, the GMRT. And once they got land in Pune and they moved in the late 80s, that space became available. But since it was kind of allocated to TIFR, what IIC said was, why don't you guys, the new center, which you are trying to form, so why don't some of you move into this space, because you clearly lack space in TIFR. And so some of them actually moved to this center where previously Govind Swaroop and GMRT folks were. And they started working out of this makeshift space on the IIC campus. And finally, in 91 is when finally the Karnataka government gave them space on the University of Agricultural Sciences campus, the GKVK campus, the UAS GKVK campus, where they finally got the lease for the land on this existing university. And that is where NCBS came about, this is a picture of NCBS as it looks like today. So around this time, Obaid decided to move. At this point, he was over 60 years old, as you can see, but he still decided to move from Mumbai, him and his entire lab, to the NCBS campus in Bengaluru, and that is where he stayed for the rest of his life. Yeah, and if you are interested, there are several interesting stories about these 12, 13 years of struggle that the biologists in TIFR went through to be able to set up NCBS. For some of us, it's not obvious who are not from the system. Okay, so that's all I had to tell you about him in the institution, him doing science. But like I said, Obaid was a man of many interests. Beyond the lab and institution, he actually was a huge proponent of science outreach at all levels, both within scientists and beyond scientists. So one of the things that he came up with was these fascinating seminars, which were like these Gordon Research Conferences in the US, called the Mahabaleshwar seminars. These are not held as regularly anymore. This was his brainchild, where he decided to have these retreats in this small school in Mahabaleshwar in Maharashtra, which were theme-based, wherein he would invite like some of the top leaders in some subtopic in biology, something that is very upcoming, very new, which everybody's working on, to India. These were people both from India and abroad, and some latest advanced. And these were theme-based courses for around a few days, where they stayed in this Ahmednagar College campus, and these classes were held in this chapel in this campus. And then they would go for hikes, spend all day just talking science. And there are several interesting stories from these Mahabaleshwar seminars. And these are some of from the TIFR website, some of the different topics on which these seminars have been held over the years. And there are 40 such seminars which have happened. And the last one happened in 2016. So the past 10 years, fewer of these conferences were happening, but for the first 30, 35 years, every year there was one. Religiously, they used to have one of these seminars every year. And like you can see, several of the top leaders from Stahl to Eberhard, Maynard Smith, they all visited Mahabaleshwar over these 40 years. This is a fun picture which I actually found on the Mahabaleshwar seminar, from the archives of Mahabaleshwar seminar. It's around 78. And for those of you who are in biology and know many of the current leaders in biology, it'll be fun for you to recognize some of them from this picture. So this is, of course, Obaid sitting here in the middle. But over here, you can see, for instance, sitting over here, you can see Madhav Gadgil, who many of you know, the famous ecologist. You have Somdatta Sinha over here. She is a famous computational biologist. These people have now retired. So this is way long ago, over 40 years ago. You have Shaila from ISC over here. This is Veronica. This is Nanjundia. Then you have Sameer Brahmachari. Of course, many of you have heard of him, Sameer Brahmachari from IGIB over here, and so on. So, and I guess each of you can recognize some of these leaders in these old pictures. I had a lot of fun looking through this picture. Yeah. And so beyond this, of course, there were also other short courses, other visitors to the TIFR NCBS, TIFR system in the 70s, and then NCBS later on. You see here Francis Crick of DNA fame, Alan Garren, this is Michael Sella from Weizmann, this is Morris Cox and Geller. These are all really famous molecular biologists. And this is, of course, Sidney Brenner, the Nobel laureate, visiting in 2012. This is NCBS. And all of them essentially came on visits, spent time with the students, very interesting scientific exchanges. And there are several reminiscences, like, you know, stories from people who attended these seminars and these winter schools, summer schools, telling us how these small courses completely shifted their whole experience with the sciences. He was also very active internationally, regular visitor to the ISTP in Trieste. And also he was very, and this is something I just learned, that he was quite instrumental in setting up life science research in Pakistan, in fact. So both at the FSC, which is in Lahore, as well as the CMB in the University of Punjab, he was on the advisory board helping them set up these life science schools within Pakistan. And in fact, this is a picture which again I've had fun seeing. This is a picture where he's with Parvez Goodboy, who actually visited our campus a few years ago, and Riazuddin. Both of these are Pakistani physicists, and this was in one of the first LUMS workshops wherein they all got together to help the Lahore University come up with a life sciences school. So there are several such stories of how he did a lot of international outreach, trying to help people set up their own centers, set up their own units. And even Nanjundia was very instrumental in some of these workshops with him over the years, over several years. Okay. So of course, I would not go into the accolades. He given the story that I've been telling all of you. Name it, he had it. He had the Padma Bhushan, the Bhatnagar. This is a picture of him signing the book for the National Academy, the US National Academy of Sciences, Fellow Royal Society, all of this. But what some people, of course, won't know, people who just read his papers, was that he was essentially often called a Renaissance man by people who knew him well. And that essentially meant that he was a man of many tastes. So there are several things which he was interested in, and some people said that it was almost annoying, given that he kind of knew so much about so many things. So he had a lot of interest, and he knew a lot of Urdu poetry and Persian. He was also interested in music. So during his time in Mumbai at TIFR, he actually managed to go and learn the sarod from Annapurna Devi, which by itself is a feat by itself. Even music from the Dagar brothers, and he apparently used to practice every day. His students told that he wasn't very good, but he was very, very persistent in his practice, his riwaz. Again, not surprising given his other stories. He was doing rigorous science at the bench till the last day of his life. And another thing that is very interesting and important to mention is that he had a very, very high sense of social commitment. He believed in free thinking, and this was true throughout his life. What I didn't tell you about was during his time as a student in AMU in Aligarh, he was actually a student leader. He was actively involved in politics. This was the early 50s. And in fact, there is the story about him being one of the only, or probably the only fellow of the Royal Society who was actually in jail for almost 2 years during that time when he was a student leader. I will not go into those stories of those days. They are in fact they just take up an entire day if you want to read those stories about his life. But of course, over the years, he was less revolutionary, if you will, but he was still very, very committed to social issues. And this came out in several of the conversations that he had with several of his students and colleagues. And in fact, just to acknowledge this side of him, the NCBS actually has recently come up with this Siddiqi's chair in history and culture of science, given the fact that he never restricted himself just to that domain of science. And this has just started, and you'll see that the applications were just closed. And so this was kind of one of their acknowledgements of him as this true leader in terms of all kinds of life in general. Okay. And finally, reminiscences. Again, I had a short video here, and I won't try sharing it because it might again go into whatever happened. But these are some very fun letters. So this is a letter from Vijay, which he sent to Obaid. Vijay, again, the PSA, current PSA of GOI, telling him about how, so apparently Vijay himself was also politically active as a student. He was very involved in the emergency-related activities, political activities, where he was campaigning for civil liberties. But after the elections were over, he felt very empty, is what he says, in spite of things going the way he wanted. And the way he came back and started kind of reliving was by reading this paper by Obaid while he was sitting in some office somewhere. And he kind of narrates his story over here. And what he did was, he was a chemical engineer in IIT Kanpur, and he had planned to go abroad for his PhD. But reading this paper from Obaid, sitting somewhere in Delhi, he just decided to quit those plans and move to Bombay. And that essentially was Vijay's introduction to neurobiology. And he still does work on chemosensation in flies. So this is a quote from Vijay, that science is fun, always question authority. This is very important. And again, this is a fun quote from Vijay as well, wherein at that point of time in the 80s, everyone knew the Tata Institute because of Obaid. This is from the Nobel laureate Lewis, who was in Caltech when Vijay was there as a postdoc. And he said, "Oh, are you from India, from the Tata? Do you know Siddiqi? How does one person do such wonderful work in so many different aspects of biology?" And so on. So I'll share this with you, and you can watch these videos also, which are related to Obaid and how he was this iconoclastic molecular biology guru, which some people called him at that point. Okay. I guess, yeah. So finally, I thought I'll end with this slide. So all of this was, of course, telling you stories about people remembering Obaid, how he was, how they influenced their lives. And I thought I'd put in a personal touch, which is essentially the fact that I never knew Obaid at all. However, as a student in BSc, MSc, I'd heard about him. And in those, even at that point, this was late 90s, early 2000, he was considered this image of him was there. He was kind of this bigger than life person. Literally also, he was like 6.5 feet tall. And all of us were in awe of this figure. And it so happened that I'd actually gone for this conference to NCBS. NCBS Bangalore had just started. And all of us participants were, will we get to see Obaid? Will we get to see Obaid? And we all thought that he would be this very stodgy gentleman sitting in his office, and we would probably see him from a distance and run away and so on. And we were all standing in the NCBS cafeteria waiting to get our lunch or breakfast or something. And we suddenly see this tall man standing in line with us. This is not my picture, by the way. This is from the NCBS archives, but I just put it out there. And everyone told us that is Obaid. And I've later read about papers, and this is of course part of the NCBS culture, which is very inclusive. There is much less hierarchy than you see in Indian academia, where people sit at the same bench in the cafeteria, chat, free exchange of ideas. Everyone calls the faculty by first name. Obaid was Obaid to all his students. And we were like, literally, we froze in our place, because here is this guy we've all heard about. And like, you're in your early 20s, you're literally in awe of this guy. And he's just standing there in line with you with this tray, which we all had. And I guess and then what happened was even crazier. We were not even NCBS students. We were sitting at this kind of table, having our lunch, and he came and sat with us. And of course, all of us were very stupid. We just completely froze and we gave silly smiles. And he started asking us what we were there for, why we were there. And then he kind of moved on to the next table because there were several participants in this conference. But if you listen, you hear stories about Obaid, that was essentially Obaid in his prime. He was always there in the cafeteria. That was his favorite place, with a cup of tea, hanging out with students, no matter who they worked with, and just talking science. And so I'd like to stop with this picture of him in the NCBS cafeteria, where apparently he spent a lot of time chatting. And I would like to thank you for your attention. And before I stop, of course, I'd like to acknowledge some people. These are some of the sources I read on the internet. This is a picture of Obaid, which his students came up with after he passed away in 2013. But I would like to give a big shout out, huge shout out to the NCBS archives. I had heard about, but I didn't know about the archives. But this is a fascinating resource, not just about Obaid, but about Indian biological sciences and sciences in general. It's online, digital, but it also has a physical presence. And very appropriately, it's housed in Obaid's old lab, which is old lab and office, which I show over here. And a special thanks to Venkat Srinivasan, who kind of heads the archives. And if you have time, do it. It's a time sink. You can just go there and get lost. And like I said, it gave me a good way to run away from the reality of the world now for the past few weeks. I've been reading a lot of interesting papers there, articles and pictures and audio clippings and so on. And also big shout out to Professor Nanjundia. So Professor Nanjundia actually visited us a few years ago to give a talk. And at that point, I didn't know of his close association with Obaid. And it turns out that they were really good friends. And after Shudipto approached me to give this talk, I actually wrote to him, and he was very, very helpful. So he actually pointed me to some of these sources which I read. And Nanjundia even did edit this special current science volume in memory of Obaid after he passed away, which is a very, very interesting read. And I also list that over here. It's on the Current India website, Current Science website as well. And finally, of course, all these other sources about him. And thank you. Thank you for listening, and sorry for the technical difficulties. And yeah, I would like to point out this YouTube video from which I was showing that excerpt. That is essentially the TIFR produced video on Bhabha in general, and which has a lot of these little snippets on Obaid's role in the TIFR story, the TIFR Mumbai story. Okay, thank you for your time, and I would be happy to answer any questions.